Thank you for coming to this presentation. I would first like to thank all of the organizers for for inviting me to give this talk. My name is John Baxter. I'm part of the Medicis team at the Laboratoire de multi-disciplinary de l'image. And this project, done in collaboration with Alfonso Estudillo Romero and Pierre Janin, was funded by the region of Brittany, as well as the Rennes Institute for Clinical Neurosciences. I'd like to call this talk A Tale of Two Brains. The first brain is that of the patient, and this brain is ill. It notices that its body is shaking, it's having trouble moving and maintaining a correct posture. It also notices that it's harder to think and also to sleep. This brain might have Parkinson's disease. Now, Parkinson's disease is caused by the degradation of a small region in the center of the brain called the substantia nigra. And this degradation leads to a cascade of effects all over the entire brain. So this human brain is sick, and our computer brain actually knows. This is because our computer's brain uses a technique called convolutional neural networks. Convolutional neural networks are a mainstay of deep learning. They're when you use a type of image processing operation, the most basic type actually, called a convolution, and apply that to an image repeatedly. So even though each convolution is very basic and very simple in itself, by applying different convolutions at different times, we can actually extract some very abstract and more complex information, such as whether or not a particular image belongs to a patient who happens to have Parkinson's disease or not. So by training this neural network to distinguish between people with Parkinson's disease and people without, that is, healthy controls, we can create a computer that can automatically diagnose these patients using different types of medical images. The types that we use are called diffusion tensor images, or DTI. So we have a human brain that's sick. We have a computer brain that can tell. Thank you for listening to this presentation. No, it's not terribly fulfilling. I agree, and the presentation's not over yet. It's really not enough just to have a machine learning tool that can diagnose a disease. What we really need is to understand how that tool works. And this gives us some insight into how that disease works. Instead of only looking inside the patient's brain, we should also open up the computer and take a gander inside its brain as well. Let's see what the computer is really thinking. This has to do with the development of something called imaging biomarkers. An imaging biomarker is a specific measurement of a specific region of an image that correlates either with the presence of a disease or with its progression. Now, in traditional neuroimaging research, what happens is an expert neurologist chooses particular measurements and particular regions, and then we use traditional statistics to tell if those measurements really are imaging biomarkers. But machine learning is completely different. In machine learning, it's the machine itself that determines what parts of the image are interesting, and it's the machine itself that determines how to measure those parts of those images. Unfortunately, though, machine learning is largely known as being a black box. So how do we look inside? The framework developed by Alfonso is actually very elegant. That is, we use the same mechanism that the computer learns by, backpropagation, to also determine what the computer is most interested in. That's called a sensitivity map. So if we get a sensitivity map for each patient, we can then register all of the patients and all of these maps together into a single standardized space, and then tell what parts of the images are consistently picked up by the computer as being interesting, as being either positively correlated with Parkinson's disease or negatively correlated with it. This has given us some very exciting results. The first is that we can interpret these maps largely in terms of the ways we interpret diffusion tensors as a whole. That is, our computer picks up that some regions are interesting because of an increase in the amount of diffusion, and other parts are interesting due to how that diffusion is shaped. That is, whether it follows particular lines like fibers, or if it's more diffuse like open fluid. Because we don't tell the computer exactly where to look, it's free to look anywhere in the human brain, and that has also led to some really interesting results. Specifically, one of the strongest biomarkers that our algorithm has discovered largely has to do with the cerebellum. Now, this is far from the region that degrades in Parkinson's disease, but it's really, really highly related to movement and motor activity. What that tells us is that Parkinson's disease changes networks, even far from the regions that are degraded. And it's the change in those networks that leads to those motor symptoms. Another interesting thing is that our computer automatically determines what we like to call non-local biomarkers. What this means is that the computer has found different measurements in different regions of the brain, and it's the correlation between those measurements that is correlated with Parkinson's disease, rather than any of those individual measurements on their own. A useful example of this is asymmetry, which is when you take the same measurement to different but corresponding regions of the brain, one on the left and one on the right. And it is the difference between those metrics which tells you something about Parkinson's disease, whereas each region individually might not. And this is really important because Parkinson's disease is lateralized. It starts on one side of the brain before the other. So our network can actually automatically tell what lateralization, that is, what side of the brain the disease originates from. So we now have a network that can distinguish between patients with Parkinson's disease and healthy controls. However, there's a catch. Our database is largely constituted by people with relatively late stage Parkinson's disease. And what we really want is to be able to catch early stage Parkinson's disease. But Parkinson's is not monotonic. Early stage disease is not just a lighter version of late stage disease. So right now we have a network that can tell patients from controls, but the real question is, can we make a network that tells patients from each other? This is where using machine learning can really help, because we can add different outputs to our network. That means that our network can detect different things. So instead of having the network predict whether the image comes from a patient or from a control, instead we could always give it images from patients and have it predict how severe that patient's symptoms happen to be. And we can use exactly the same approach as we had before. That is, we can use the same backpropagation algorithm to determine these sensitivity maps and register them all together to see what areas of the brain are positively correlated with symptoms and what parts are negatively correlated. This has been really exciting because it gives us really different biomarkers than what we had discovered before. As I said, Parkinson's disease is lateralized. So when we're comparing Parkinson's patients versus controls, our biomarkers for that were also lateralized. What's interesting is that our biomarkers for progression are lateralized, but they're lateralized on the opposite side. As Parkinson's disease progresses, what happens is it moves from one side and spreads into the other, meaning that originally your symptoms happen on one side of the body and then they move to happening on both sides. So a good marker of progression is actually telling if the disease has moved from just being on one side of the brain to being on both, which means that the biomarkers that we discover actually really correlate well with how we understand the progression of Parkinson's disease and its symptoms. So what we've got at the end of the day is not only a neural network that can predict whether or not a patient has Parkinson's disease, but can explain why it's making that prediction. And we also have another network that can explain why it's making a prediction of how severe that Parkinson's disease happens to be. And all of this is based on diffusion tensor images, which give us information about the diffusion that happens in the brain. So this is in things like white matter fibers or the cerebral spinal fluid, such as ventricles or sulci. Another interesting point is that the system that we've developed is very general. It can be used for other neurological disorders. So right now, we're expanding this research not only to look into other movement disorders, such as essential tremor, but other completely different neurological disorders, such as dementia or even drug addiction. But there are limitations. The most important one for us is that this network really shows us everything, everywhere, all at once. The network is showing us biomarkers for potentially different aspects of the disease, but it's showing them all at the same time. That means that it takes a human expert like a neurologist to actually tell how these biomarkers are related to the disease itself. We found a bit of a way around this. If we ask questions that are a little bit more difficult and specific, like predicting symptoms instead of just whether you're a patient or not, we can get biomarkers that are sparser. That is, they light up fewer areas of the brain, and that makes them easier for us to interpret. The other reason why I chose this everything everywhere all at once is it fundamentally uses a population perspective rather than focusing on individual patients. This means that it mixes together different possible subtypes of the disease that should actually be distinguished from each other. This is especially important for neurological diseases that we still don't fully understand. And thus ends our tale of two brains, a human brain that happens to be sick and a computer brain that happens to know why. Thank you for your time and your attention.